which means we want to know what you have done, how you have carried out the project, and how it has impacted, meaning the conclusion about the project. So that will drive others to want to do more or do better. It's either we fortify or we renew. So, and each presentation is not going to take more than five minutes. 5 minutes. Yeah, because of time. Some people are still going to a far place. That's why I'm telling us now that we should try and arrange ourselves. Do less of introduction. Do less of introduction, because your work that you have done is what we are interested in, and the results. And if there is a problem that you want to recommend for us, tell us. We have researchers in the house that can go back and work on it and still come back next year and present. So that is what we want to do. The first presenter here. We don't want to waste our time. We will need a timekeeper who is going to help us with that. 5 minutes, so that we'll be able to, it's like we have the highest number of presenter here. We need two timekeepers. If one is not coming, the second one will appear. 12:57 now. Okay, thank you, ma'am. The first presenter. Let them come up. I think they know themselves. Let me see if I have it on my own. Please help us to project the presenters there. And I believe they've already given their slides. The information getting to me now is like we're going to have one room for this presentation. The projector there is nobody to operate them. So, and some of our presenters are not available. So we will try and manage our time. That's why I said, in short, I have not heard of this, but I've been looking at the way things are. 5 minutes per person. I think we should be able to accommodate those who are on ground. The first person on this one, this background. Yes, everybody. Aseba et al. That is optimization of maize and pigeon pea flour blend for ukpo production. They just wrote ukpo. Okay, ukpo oka production. Okay, you are here. Thank you. God bless you. 5 minutes.
Yes. Okay. Good afternoon, distinguished ladies and gentlemen. My name is Asieba Lydia Nkema Konam, and I'm here to make a presentation on the topic optimization of maize and pigeon pea flour blends for the production of ukpo oka. Next slide, please. Next slide, please. Okay, this presentation outline. So by way of introduction, ukpo oka is a maize-based pudding similar to moi moi, a bean pudding. So it's popular for breakfast and lunch in the southeastern parts of Nigeria and also in Delta states where I happen to come from. Ukpo oka, being a maize-based pudding, is typically low in protein and it is often sometimes consumed without any additional protein sources. Next slide. Protein energy malnutrition is a major health challenge in developing nations, and this is as a result of inadequate diets and excessive reliance on single food items, leading to monotonous diets. So cereals, we know, are limited in they are limiting in essential amino acids like lysine and tryptophan. So blending cereals and legumes to create more nutritious food options has been suggested as a cost-effective approach to combat PEM, the protein energy malnutrition. Next slide. PGP, it's a highly dense nutritional legume. It's called, it's known as with different names, like otili in Yoruba, we call it fiofio in Igbo. So next slide. Next slide. So traditionally, ukpoka has been produced from maize. When maize is in season, that's when ukpoka is eaten. So this study is looking at blending, enriching maize with legume and using pigeon pea being an underutilized legume in these parts of the country. So we want to enrich ukpoka with pigeon pea. So the main objective of this study was to optimize maize pigeon pea flour blends for the production of this meal, and using high optimal mixture design under the framework of a response surface methodology. Next slide. Materials and methods. Freshly harvested maize, pigeon pea were purchased and then processed. Processing of maize flour as described by Atsuma et al. Processing of pigeon pea flour method described by Atsuma et al. Next slide. This is the flow chart for the production of maize flour and then the production of pigeon pea flour. Next slide. These are the samples. The sprouted, we have the maize grains, the maize flour, then the pigeon pea seeds. That's a picture of the pigeon pea legume. Then the sprouted pigeon pea, the hulled, and then the flour. Next slide. So, like I said, this work is actually on the optimization of the maize flour and the pigeon pea flour. So the experimental design, high optimal mixture design approach within the framework of RSM was employed. The total maize and pigeon pea proportion was 100%, and this means that we have maize proportion in the blend of between 50 and 80%, while pigeon pea proportion was between 20 and 50%. The independent variables were the mixture components, that's the maize flour and the pigeon pea flour. Then this gave us 13 experimental runs, of which out of these 5 were repeated runs. These runs were then formulated and analyzed in the laboratory. For this, we analyzed the protein content, ash content, and the iron content. We selected this for optimization. Looking at the protein content, we want a product that is high in protein because of the amino acids, the essential amino acids in pigeon pea as a legume to complement the maize flour. So iron content was also selected because it has been found out that pigeon pea has considerably high amounts of iron. In fact, it's been used in some studies have shown that it's used even in the treatment or management of sickle cell anemia. So iron was also used as a response. Then ash content for the total mineral content of the product. So I selected the ash content. Next slide. Next slide. So the experimental results were entered into the design expert software, and the software fits this response data into a polynomial equation. That's the equation, and this equation is just showing us how the changes, how the effects of the independent variables, what they have on the responses. The responses were the protein, the ash, and the iron. So this is the equation. Now models were generated and evaluated statistically using ANOVA, still in this design expert. Okay, next slide. after which a numerical optimization was done. This was employed to determine the optimized factor conditions. Now the independent variables were kept within range, while the goal here was to maximize each of the responses, the protein, the ash, and the iron. Optimization was achieved using a desirability function, and in the end we had a desirable value of, next slide please, 0.605. Next slide. Next slide please. Okay, so this is the result and discussion. The impact of blending maize flour and pigeon pea flour on the responses are shown in figure 3. We'll go to that. Now it was noticed that as the amount of pigeon pea flour increased, there was also a significant increase in the responses, that is in the protein, the ash, and the iron. This is expected. So all response models had significant p-values of p less than 0.05, and they had regression coefficients ranging from 0.9 to 0.947. This implied that the model could explain 90 to 95% of the overall variations in the response values, and thus these models were fit for prediction. Next slide. An optimal maize pigeon pea blend of 54.3% maize and 45.7% pigeon pea with a desirability factor of 0.605 was achieved after subjecting the predictive models to a numerical optimization, as also shown in the figure. You will get to the figure. This optimal blend had a corresponding protein content of 24.31%, 1.45%, and 5.03 milligram per 100 gram for the protein, ash, and iron concentrations respectively. So the optimal formulation was further analyzed by conducting laboratory analysis to validate the optimization, whereby when we got a close range to this, after analyzing it, it validates the optimization that the response models were effective. Next slide. So this is the result, experimental runs, the maize, pigeon pea, the different formulations, and then the result from the analysis. Okay, the next slide. Next slide, we have the next slide. So these are the plots. The design experts, he gives this contour plots showing, this plot actually shows how the changes in the proportion of the maize and the pigeon pea flour affected the responses, how they influenced the responses. This is for protein. We can see that the increase as the proportion of pigeon pea increased in the flour, in the formulation, the protein content also increased, and we can also see that in that of the ash and then of iron. Next slide. This is the effect of maize flour and pigeon pea flour on iron response, and the plot showing the desirability factor 0.605. This means that the closer it is, the desirability factor, the closer it is to 1, it shows that the response models is fit for optimization, and we had 0.605, so it makes it fit. Next slide. So in conclusion, this study has provided an optimal blend of maize pigeon pea flour expected to have improved nutritional profile for the production of ukpoka. By incorporating pigeon pea flour into the maize-based formulation, the nutritional profile of the flour blend was significantly enhanced. The use of RSM enabled a systematic evaluation of the interactions between maize and pigeon pea flour proportions, providing a predictive model for optimal blending ratio. Further studies should incorporate functional and sensory evaluation to ensure that this optimized blend meets consumer acceptability. Thank you. Next slide. These are some of the selected references. Thank you.
Thank you very much. Any question, please? Question. If you have any question for her for what she had delivered to us this afternoon. Okay, sir. Thank you, sir.
Sorry, a good presentation, and like we have seen this. My question is just very simple, and it has to do with the idea you have in blending this to have a product. How do you intend to get this product to the market? What are the arrangements you are making to get it to the market? Like we all concluded here, the era has gone when you have research, microorganism do this, microorganism do this, and it's just there under the shelf. How do you get microorganism out to go and help somebody somewhere? Thank you.
Thank you, sir. Well, this product, it was prepared, and after prepared, it was packaged. I believe it could, at least when shelf life studies are done on it, we could see how long it will last. So it's something I feel can be commercialized, because we notice that, like in my place, upoka is majorly consumed when maize is in season, because it's being consumed using fresh maize corn. But in the process of doing this, I realized that we could have it, the maize in dried form, even after you've purchased the maize in the wet form, processing into flour, having it in dry form, in shelf-stable form, and at the end of it, sensory evaluation was done. I mean, it still tasted, people that are familiar with upoka said, yes, this tastes like it. So I see that you could have it all year round, not only when maize is in season. So packaged in, I mean, when you package it in materials that would preserve it, and it's in dry form already, I feel it can be commercialized. So, and having it now enriched, not just maize alone, at least you are able to, you can tell that what you have is a complete meal. You have your maize complementing the amino acids from your maize, complementing that from the legume. So yes, I hope it should. Okay. No, not 45. The way it is, the flavor.
I know. It has been there since before we were born.
Thank you very much for this presentation. And I like this presentation because it's aligning with the theme. You know, government is looking at, when we keep saying we are the professionals, what are you doing different? A lot of products want to come into this country. They say they want to feed our IDPs. And you know, Nigeria is so blessed. We are here. You know, there are regional, like now, this product now, I was just thinking about it. It is something that can be made and recommended for feeding the marasmic children now, you know, malnourished. But I just want to be specific, because marasmus, you know, kind of like, if we have things, we are doing a lot of things like this, I think we should, I don't know how we can publicize this type of thing. Let people pick it, because I remember those days we do research. For me, I don't eat ogbono again based on analysis I did on ogbono, and I discovered the fatty acids there are not healthy. On my own, I just withdrew. The one for ube is excellent. How we incorporate, these are natural ingredients. I just want to recommend that this type of thing we are doing here, we just gather it and let it pass through Federal Ministry of Science, and you know, they've added innovation now. Yes, because what we are doing here should not just be when we finish and then we go. I listened to the objective of this particular research and I followed it keenly, and I said, well, it should be us included. Particularly the world is thinking, they are doing as if they care more about our problem than we ourselves. Whereas I know we are doing a lot. So this type of thing should just be collated at the end of it. Let's not just go silent. Thank you very much.
Thank you.
Thank you very much. You can step down. Thank you, ma'am. The next presenter, please. Approximate compositions of millet maize.
Good afternoon, distinguished member of the high table, my respected colleagues and ladies and gentlemen. My name is Kole Okpoluwafunlola. I want to present the research study carried out by myself and my team with the title Proximate Composition of Millet Maize-Based Complementary Winning Foods, fortified with African breadfruit, banana, milk, and soybeans. Winning, next slide please. Winning is the introduction of food or fluid to infants in order to meet up with their nutritional requirements as they are growing. But complementary feeding is a very sensitive aspect of bringing up children. One, because of the safety in the food and the composition in the food in order to give them the nutrients that they require for their proper growth and development. There are many importance of complementary food. It gives proper growth and development to the infants, and it prevents malnutrition, and they are very, very cost effective and affordable. The raw materials for their production are always available. Therefore, it is better to prepare these fish from the locally available raw materials. Next slide. Problem statements. Protein energy malnutrition among infants and young children remains a major public health concern. Traditional cereal-based winning foods are often low in protein and micronutrients. Therefore, it is needed to develop nutrient-based complementary foods that will be able to provide all the nutrients that these infants need for their proper growth and development. Next slide. Aim and objectives. To evaluate the proximate composition of millet-based complementary winning foods fortified with African breadfruit, soybeans, banana, and milk. And the major objective is to formulate complementary winning food blends to determine the proximate composition of the formulated samples, and then to compare the quality in terms of nutrients of the different formulations with standards. Next slide. Materials and methods. Raw materials used were millet, maize, African breadfruit, soybeans, banana, milk, and powder. Chemical reagents were used for the treatment of the materials in order to prevent browning and bitterness in some of the materials. Then the analysis was carried out using the standard method of analysis. ANOVA was used for the data analysis. Next slide. Experimental designs. Finger millets is a common raw material that are not well utilized in Nigeria. So all materials were treated by sorting and cleaning, and then the millet was sprouted until 2 centimeter pneumo came out. Then they were then washed and dried at 60 degrees using cabinet dryer. The dry millet was then milled, sieved, and then we got the flour out. Next slide. Maize banana flour. The maize were washed, dried, and then milled into flour. But what we did was that because of the nature of banana, it has high sugar content. So in our test running, banana refused to come out from the miller. So we had to mill the banana with the maize. So we now form formulation that comes out with maize banana flour by blending the two of them together before now using them for the complementary formulation. So the two of them were dried and then milled together, then sieved together to give us our maize banana flour. Next slide. Soybeans flour. We made use of the light yellow soybeans. After sorting the soybeans, they were soaked in the sodium hypochlorite in order to remove the bitterness in the soybeans. Then they were then steamed for blanching for 20 minutes, dehulled and dry, then milled, sieved into flour. Next slide. African breadfruit flour. The African breadfruit, some people say that it's very, very expensive. In my village, African breadfruit happens to be a waste, serious waste in my village. So we know Yoruba called it afon, and the Igbos call it okwa. Ukwa. So the breadfruit was cleaned, parboiled for about 60 minutes. Then after cooling, we dehull the breadfruit into 8% moisture content, roasted and milled the breadfruit, then sieve it to get our flour out. Next slide. This is our formulation. we came out with 5 formulations from our blends. Then this is the banana maize. We use 30% of a maize, banana maize, for the first sample. Then just see it as it is on the table in case because of the time. Then the banana maize flour formulation, we use the 4.2 to 28.6% of banana mixed with 3 grams, mixed with 71.4% of maize to get the formulation. This was what I was saying initially, that we blended them together and later got the percentage that we needed for each formulation. Next slide. These are the analysis carried out on the blends: moisture content, crude protein, crude fiber, hash content, and then crude fat, carbohydrate content. We now use the method of AOC to analyze the data. Each analysis was carried out in triplicates, and we found out the mean value of each of the results. The mean value was now subjected to ANOVA to get the differences. Next slide. From the results, where is the result slide? This is the moisture content from the results. The moisture content range from 2.78 to 2.92. Then the protein content from 13 to 13.21. Fat content from 12.14 to 12.37. And ash content from 2.74 to 2.90. And then if you look at the result of this table, because after getting our blends, our formulation, we now compare each of them to one another, and then compare the overall with standard, the RRDA and the protein advisory group. They go for a standard for complementary feeding, that we have to have a limit to the moisture content so that the product will be shelf stable. Then the protein content, which we know that the infants, they need much protein in their diet, so we follow the limit. Next slide. These are our results. Across, we can see that for the moisture content, there is no difference in the mean value obtained. And according to RDA said that moisture content should be less than or equal to 10%. And our sample gave us the range of 2.79 or 6.78 to 2.92, which is very, very good for the product. And looking at the protein content, RDA said it should be between 15 to 20%, and PAC said that it should be between 15 to 20%. And our result gave us 13.0, and then the highest was 13.21. Though the protein content was very, very low to the standard, but they are still within the range and can still be acceptable. Looking at the crude fats, RDA said that it should be between 10 to 25%, and PAC said it should be between 10 to 25% as well. And our whole crude fat gave us 12.14, 12.37, and then the lowest was 12.14, while the highest was 12.37. We can see from the range be given to us by the standard 10.25, we still fall within the range from our results. The ash content should be less than or equal to 5%. Our hash was between 2.74 and 2.90, which I still be within the range. And the next one is the percentage crude fiber. Standard they said that it should be less than or equal to 5%. Our result was within the limits. Then percentage carbohydrates, we fell within the limit because the highest limit is 75%. And our own, we have 75.48%, which you believe, 78.08%, which you believe that they are still okay. But the result of the ANOVA means with the same superscript within the same column and not significantly different. That is when P is greater than 0.05. For those that have carbohydrate, we have difference in the carbohydrate content because of the millet and the maize that we used. though it is still okay and the results still fell within the limits. From these results now, you can see that our product is still okay for an alternative to conventional complementary winning food, because all these materials are locally available. The only difference in this our result is the protein content that is a little bit lower to the recommended standard. But most of Nigerians and the other our mothers, once we are preparing our baby food, we still add milk to our baby food, which I believe can still boost up the protein content required by the children. Now our next slide, please. Like I discussed earlier, that the moisture content was very, very low, and it was okay for the complementary food, for the shelf stability, and then for the texture of the product. And then the African breadfruit, soybeans, and milk contributed greatly to the protein enhancement, because BCE among the stable has the highest 13.21%, and BCE had the 13%. However, the standard was asking for 15%. Though we are still okay with it, because we are still going to add milk to the baby food. African baby improved nutritional diversity, because this product is rich in vitamins, mineral, is very, very rich in protein, and accept a little carbohydrates and little fats. And then we have banana, which enhance the flavor and the carbohydrate content. Fortification improved the protein, and results agree with the previous studies on the fortified cereal legumes. From the research or the previous one that has been done, we realize that our product came out better than other studies in the past. So the next slide. In conclusion, the formulated complementary foods showed improved nutritional quality. The formulations can help reduce protein energy malnutrition among infants, and the formulated product has the potential to contribute to the improved infant nutrition. They may serve as alternatives to conventional or commercial complementary winning food. For this, we are now advising that complementary winning food can be locally prepared from the locally available materials, and since they are always available and they are always accessible, we can make use of them. It's only the way that we prepare them. We realize that in the olden days, the normal winning food that we normally prepare is merely maize-based winning food, which is very, very low in all the nutrients for this infant. But if we try to fortify this maize-based food with all these materials that we made use of, they will have better feeding for our children. Thank you.
Thank you very much, ma'am. Any question? Thank you.
Thank you very much for your presentation, ma. My name is Jane. I'm a clinical dietitian nutritionist. I want to appreciate you for your presentation. My number one question, please, where is your village? Let me come and pack the okwa. Okwa is expensive. I will come. I'll come and pack it. It's highly nutritious, and it's very scarce now. So thank you. And again, I have been a strong advocate of our local foods. People that know me know. I find our local foods even for complimentary. So this is very commendable. But I have something to point out. You see, I feel that the combination of foods are too many. Let me explain. Now, we prefer, if you want to make complementary food for children, we are talking about pediatric nutrition now. You should just use like highest 3 products. You know why? So that if the child is allergic to something, you point it out. Yeah, because when you are talking about the physiological composition of the child, you know they are very delicate, and the body will be struggling to break breadfruit, break soybean, break banana, break this, break that. It can actually lead to complications which even the doctors will not detect, and the child will be so uncomfortable. I've handled a lot of children like that. You know, what the doctors will end up giving them is maybe like a purgative, antibiotics and all that, but you don't know that it's excessive combination. So I will suggest, for example, I've been working with soybean for years now. Soybean has about 33 grams of protein per 100 grams, which is okay. So if it's me, if I have to do this, I will do soybean, millet, the banana and maize. Then I'll now use the African breadfruit for, add it to maybe the another food like acha or sorghum, and make another food. So that's my suggestion. All in all, I enjoyed your presentation. Thank you, ma'am.
Yeah. Good day, everyone. Yeah, my name remains Okechuku. My contribution is on soybean. Soybean has many anti-nutritional factors. So I don't know, during your processing, how you took care of that. Yes, okay. Then, in addition to what my sister said, please, when we are handling kids' food, we consider their physiological structure. You understand? Now, and when it comes to protein, there are numerous types of protein. There are those that their bodies cannot accept, those that can upset them and to do all sorts of things. So, and in the presentation, I expected to hear maybe a kind of a clinical testing. Please, it's necessary. We are food scientists. We just end up in. Please always do that clinical testing before it can be acceptable in the market. Thank you very much.
I want to respond to some of the things he said. The clinical testing is part of our research. We are still going to carry out the testing on the products. And then about the soybeans treatment, the soybeans was soaked in the water, and then it was blanched before peeling. And then the heat treatment during drying too will take care of some of the additional factors. Thank you, sir.
Thank you. My name is Wumi Fatumbi, CEO of Jisubo Ventures. I'm an entrepreneur. And what I look out for is products like this, how can it get to the market? I'm also very conscious of cost. I believe in a product like this, maybe you should exclude milk and see the nutritional value, because milk is expensive. And you are already using products that are high in protein. Must you add milk? That's my question. If not, can you, because you have to also think of cost at the end of the day. Okay.
Why we added milk to our product is that we believe milk will contain little fats to add to our fat that we need in the complementary food. At the same time, some parents, they find it difficult to buy milk, to add the milk to the winning food. Let us add little milk so that the child will not be lacking in some of the some vitamins or minerals that they need for their development.
I will suggest we don't add milk at the production stage for children that are lactose intolerant. Thank you.
I want to appreciate what you have done. It's a lot of work. And just like it has been said, the many products that you kept together is going to make it very expensive at the end of the day. Because as you are doing the research, you need to also think of commercialization. Just like the industrialist said, it's going to give them some issues. Then another thing I noticed in this topic is that you are using two words that are the same. When you say complementary, that's the term we use now. We don't use winning again. Once you have complementary, you struck off the winning. It's already implied in the topic. Then when I saw millet, maize, both are cereals. Yes. So when you are talking of complementary food, you look at cereal and legume. Though you added some legume in this, but because the major raw material for complementary food should be cereal and a legume. You understand? Apart from that, what other people have said, it's also suffice. I don't want to repeat what they have said, but reduce the number of raw materials inclusion so that industrialists will be able to fly with it. Well done.
Well, lastly, this project, if it's still ongoing, I want you to look very well into this shelf stability. Very, very important. If it's going to be given to children, you are saying you are putting milk because of fat. Are you aware that, I know you are aware that maize also has fat. Millet has fat. So that milk is soy milk has fat. So you have enough fat in all those bases. Yes. Though the components of their fat may differ, but they still have fat. Because that milk is really giving me headache from the title. Thank you. Well done.
So to add to what she said, we are doing the shelf stability. We are feeding the food to rats to see their response to the food. At the same time, we are going to do the acceptability of the products. Thank you.
Thank you very much. The next presenter, Oluwaseun H. et al. Will be presenting impact of heat treatment on antioxidants and clonesterase inhibitory activities of some Nigerian plants. Are we ready? Thank you.
Good afternoon, the high table, colleagues, ladies and gentlemen. My name is Ademuyiwa Oluwaseun Anna of Food Technology Department, Federal Institute of Industrial Research, Oshodi. I'm making a presentation on the impact of heat treatment on the antioxidant activity and choline esterases inhibitory activities of some Nigerian plants. That's Cucuma longa, Piper guineensis, and Capolobia lutea. Next slide, please. Next slide. Okay. Food processing, as we are aware, significantly affect the quality, safety, storage, and characteristics of the final ingredients of our food products. And among some of the various processing methods we use, heat treatment is one of them, and this heat treatment encompasses techniques such as steaming, frying, boiling, and all of this process triggers chemical reactions that can compromise, enhance, or modify some of the bioactive compound of our food and plant materials, and the effects vary depending on the plants, and it can either be beneficial or detrimental. And as food products are affected by heat, and to some extent, spices and vegetables are particularly susceptible to heat treatment, and with varying outcomes, varying from positive to negative. So this study investigates the effects of heat treatment, that's steaming specifically, on these three Nigerian plants, two of which are spices, that's turmeric, that's Cucuma longa, West African black pepper, that's Piper guineensis, and one vegetable-like plant, that's cattle stick, Capolobia lutea. Next slide, please. Next slide. Okay, these are some of the plant material. We have turmeric, which is a spice used as food preservative or coloring agent, and its bioactive compounds are responsible for several therapeutic properties like anti-inflammatory, antibacterial, anti-HIV, and even antioxidant, anti-carcinogenic, and anti-neurodegenerative. West African black pepper, which is we call it uziza, and iyere in Yoruba, it has thermoregulatory, antioxidant, and antipyretic activity. And cattle stick, which is called oshunshun in Yoruba language, it's used by the traditional medicine practitioner as aphrodisiac for enhancing libido, improving sexual performance, and it also has anti-neurodegenerative activity. Next slide, please. So this is our methodology. This is the investigated materials. We had fresh turmeric, West African black pepper, and fresh leaves of Capolobia lutea purchased from a market in Ota. The plants were sorted and cut and divided into 2 portions, and 1 portion underwent steaming for 5 minutes, and then it was dried, while the second portion was just dried. It was not steamed at all. And after drying, the plant material were milled using hammer mill and then packed and stored for further activities. Radical scavenging activities were done using, we used DPPH and ABTS method for the radical scavenging assay, according to method described by Kim et al. Next slide, please. Next slide. Okay. Next slide. The ABTS radical cation scavenging activity were also determined using the method by Ray et al. And the ferric reducing antioxidant power was determined using the method of Benzie and strain with modification. The choline esterase inhibitory activity were carried out using the colorimetric method of Ellman et al. Next slide, please. So these are results and discussion. From the table, we can see that from our DPPH activity, we could see that the subscript 1 represent the heat treated portion, while the subscript 2 is the raw one. So we could see from DPPH and ABTS assay that each treatment significantly increased the antioxidant activity of turmeric. In fact, up to about 10-fold. And also the ABTS activity was also significantly affected by each treatment. But for Capolobia lutea and Piper guinensis, the each treatment reduced their activity significantly. Next slide, please. Next slide.
Next slide.
Okay. Table 1 showed the effect of each treatment on the antioxidant activity, and it was observed that steaming significantly influenced the antioxidant activity of the studied plant material. The effect varied across this plant species for the antioxidant assay. For turmeric, steaming had the overall positive effect on the antioxidant activity. In fact, the DPPH radical scavenging activity increased by 10-fold, from 5.83% in raw turmeric to 52.21%, which indicates that steamed turmeric is a more effective radical scavenger. And also the ABTS assay was also very improved due to steaming. But for FRAP activity, there was really no significant change, meaning that steaming does not affect the reducing power of turmeric. While for this DPPH, improved DPPH activity could be due to the extraction of curcuminoids. We know that turmeric contains curcuminoid, that's your curcumin, the methoxycurcumin, and bis-dimethylcurcumin. And these are easily more extracted at higher temperature, because these phenolic compounds are bound to the plant matrix and require heat treatment for them to be extracted. Next slide, please. But in contrast, steaming reduced the antioxidant activities of Piper guinensis. It reduced it significantly. This could be due to the loss of some of the phenolic compounds as a result of the heat. But FRAP assay showed a significant increase in Piper guinensis, because Piper guinensis is known to contain iron, which is a pro-oxidant. So meaning that, and we know that iron helps in the ferric reducing antioxidant powers and the increased FRAP activity observed in Piper guinensis. Next slide, please. Next slide. Also for Capolobia lutea, there was a slight non-reduction, although there was a non-significant reduction in Capolobia lutea activity for DPPH activity, but for ABTS and FRAP, there was slight increase in the activity. This is due to the presence of iron in Capolobia lutea as well, which made it a better antioxidants in that regard. Next slide, please. Next slide. This is the result for the cholinesterases inhibitory activity, that is acetylcholine and butyrylcholine inhibitory activity. We all know that acetylcholine in cognitive activity, there is this neurotransmitter which is very, very important, and in cognitive decline, these neurotransmitters are being eliminated faster than they are being produced. So the effect of all these inhibitors is to make sure that more of the effect of these cholinesterases in reducing these neurotransmitters are reduced. Next slide, please. Next slide. Table 2 shows the effect of each treatment of the cholinesterase inhibitory activity. Acetylcholinesterase and butyrylcholinesterase catalyze the hydrolysis of neurotransmitters into acetylcholine, acetyl-CoA and butyryl-CoA. So in conditions such as cognitive defect or Alzheimer's disease, these neurotransmitters are broken down faster than they are replenished, which leads to memory impairment. So inhibitors of these enzymes counteract this process of breakdown, so increasing the neurotransmitters level in the brain, so enhancing our memory and mitigating against cognitive decline. Next slide, please. Next slide. So curcuminoids, which are found in turmeric, have been reported to have this activity, and this study observed that the steaming enhanced the inhibitory effects of turmeric. It increases significantly. It increased the effects of it has improved activity due to the availability of these curcuminoids, as we said earlier. Next slide, please. Next slide. Steaming, however, negatively impacts the activity of pipergenesis and Capolobia lutea due to the loss of some of the phenolics which are responsible for this activity due to steaming. Next slide, please. So in conclusion, this study reveals that steaming differentially impacts the antioxidant and cholinesterase inhibitory activity of these plant materials, which emphasize the role of each treatment in shaping their bioactive activity. Steaming enhances the radical scavenging activity of turmeric and its cholinesterase inhibitory activity due to the bioavailability of curcuminoids, but it reduces the activity of pipergenesis and Capolobia lutea due to the degradation of some of these heat-sensitive compounds like phenolics and piperine. So this finding highlights the need for customized processing to optimize the therapeutic potential of this Nigerian plant, meaning that different plants will require customized processing to be able to use them therapeutically. Next slide, please. Next slide. Next slide. Okay.
Okay, thank you. The last slide is supposed to be our references. Okay, thank you for the presentation. Before I will ask the audience for their questions, I have few observations on your presentation, ma'am. Number one, the presentation is too wordy. It's too wordy. I want to read, but I cannot read that volume. It's too much. Look at this. I cannot read it. You should just give us bullets. Speak to the bullet for us. Just give us that one that we will understand. All those big grammar you are saying now. If you ask anybody here, they cannot call it again. But the most important thing is give us those plants. This is what they are called botanically, English. If they have local names, tell us. Common names, tell us, so that we'll be able to follow you. But we understand as a scientist, we understand your English grammar. You shall be well. Any question?
Thank you very much. So let me try to summarize what you are trying to say here, or what you're teaching us. She's just trying to say that there are certain processes that are required to destabilize the anti-nutritive factors in certain foods, so that the nutritional values of such food or the therapeutic properties of such food will be readily bioavailable for the human body to use. Thank you.
Your slide for this presentation, if I want to help you to prepare it, I won't give you more than 5. More than 5 slides. The result will be number 1, your presentation 2, then your methodology. You speak to the results so that we will be able to cash it. And you have done a very beautiful job, because many people take all these plants, they eat it directly, especially ewuro. Oh, you need to see how people misuse that plant.
What's another name for ewuro?
Ewuro is bitter leaf.
Okay.
Bitter leaf. And we have very beautiful leaves that are very high in protein around us and very, very, very useful to our system. Not only Babalawo know leaves. Not only Babalawo. Scientists knows too. Thank you very much. Any question?
Thank you for your presentation. I've learned one thing, because I take turmeric and black pepper. What I've learned now is that if I want to introduce my black pepper into the turmeric, I should have steamed my turmeric, and I will now introduce the black pepper. Thank you.
The next presenter. Antioxidants and physiochemical properties of fruity tea from mango, orange, and lemongrass. Oshuma Kinwa GO. Available. Thank you very much. We are expecting you, ma'am. The next presenter should be gingering himself or herself. Okay, himself. Michael ML. A review on isolation and characterization of protein from avocado seed, processing methods, functional properties, and food applications. Be getting ready, please.
Good afternoon, panel of judges. Good afternoon, everyone present here. My name is Osuma Kiwa Gift Omowa Sodun, and I'll be talking on the topic antioxidant and phytochemical properties of fruity tea from mango rind, orange rind, and lemongrass. Next slide, please. These are the presentation outlines. Next slide, please. Chronic diseases like diabetes and cardiovascular diseases are linked with oxidative stress. And oxidative stress are caused by free radicals, which destroy the cells in the body. Antioxidants present in these fruits and medicinal plants are used to stop or to reduce the damage caused by the free radicals. There is a growing consumer demand for functional beverages that provide health benefits beyond basic nutrition. Next slide, please. Mango rind is gotten from mango and is rich in magniferin, quercetin, and flavonoids. Orange rind from orange plants contains phenolic compounds and vitamin C. Lemongrass contributes to antioxidant flavonoids and essential oil. Next slide, please. These are the pictures of the mango rind, the orange rind, and the lemongrass. Next slide, please, sir. Large quantities of mangoes and orange rinds are known as waste, and these fruits contain valuable phytochemicals that are usually underutilized. So therefore, there is a need to evaluate different formulations of fruity tea blends to identify the composition that has the superior bioactive potentials. Next slide, please, sir. The significance of the study is to reduce waste, to support affordable antioxidants, functional beverages, then for value addition to mango and orange processing industries. Also, it contributes to the research on natural sources of phytochemicals and antioxidants. It may also improve consumer health through increased intake of bioactive compounds. Next slide, please. The aim is to evaluate the antioxidant and phytochemical properties of fruity tea blends formulated from mango rind, orange rind, and lemongrass. The specific objectives: number one, to determine the antioxidant properties of the tea blend and to evaluate the phytochemical composition of the tea blends. Next slide, please. The fresh mangoes, orange, were purchased in Ikole market. The preparation of the mango rind, orange rind, and lemongrass powder was used through the process of Kayani et al. 2022, and the brewing is Tawa et al. 2023. Next slide, please. This is the flow chart for the processing of the orange rind, mango rind, and lemongrass. Next slide. There are 4 formulations for the lemongrass for both the antioxidants and the phytochemical. Next slide, please, sir. The blends of mango and lemongrass were brewed using a pot containing 500 ml of boiling water, then allowed to steep for about 15 to 20 minutes. Next slide, please. This is the table for the antioxidant results, and from the analysis carried out, LMR2 has the most antioxidant properties. Next slide, please, sir. For phytochemicals, we have LMR1 and 2. Next slide. The observation sample LMR2 showed the highest antioxidant activities across all parameters, and the reason is because the higher orange rind inclusion contains phenolic acid, flavonoids, and vitamin C derivatives. Then the implication is greater antioxidant potential, which gives better free radicals scavenging ability. Next slide. For the phytochemical properties, LMR1 has the highest value in alkaloids and flavonoids, while for the orange dominant blend, which is LMR2, it has the highest value in steroids and glucosides. Next slide. The conclusion: fruit-based herbal tea formulated from mango, orange, and lemongrass possess promising antioxidant and phytochemical properties and can serve as functional beverages. However, it is recommended to conduct further studies on sensory evaluation, shelf life stability, and clinical health benefits. These are the references. Thank you for listening.
Thank you very much for your presentation. We enjoyed that. Any question from the audience, please?
Okay, well done. Thank you for your presentation. How did you come about the formulation quantities? Why the choice of those ratios? And why is there none that is optimized? Okay, you optimized everything. So none was like a constant all through for comparison. Okay.
If it is RSM, I believe there should be a constant. Because Now, comparatively, we are having 2 samples, but the other 2 are now differ. So to compare the 4, it will not be easy. It's difficult to combine the whole thing. I think the experimental design is a little bit faulty. It's a good work. If you can go back and do the RSM very well, it will give you 13 runs will come out. And the one that is double, you can delete. But when you want to input it into the system for the result, you must remember that that one is also inclusive. You only repeat the results that you have had. Well, you know what I'm talking about. So that is the way to go about this. It's a very good work. But the only fault I see there is the methodology. Thank you. Any other question?
Okay. Thank you very much for this presentation. It's very interesting. I'm happy you brought this up, because we have a lot of waste from our food that are highly therapeutic that we don't know about. Orange rind I've been using, and it actually, I saw some research that it helps to balance hormones, and it also helps the guts bowel movement, and for some sort of cognitive ability as well. So when you combine it, I was expecting you to add pineapple rind, because most of the bromelain in pineapple is in the rind, just like most of the nutrients in orange is in the rind. So I was wishing if you could add that. Honestly, you have wonderful therapeutic tea. Thank you.
Okay, thank you very much for your presentation. When I saw the topic antioxidants, in fact, I was very happy, because all of us here, you can bear me witness that even if it's malaria you have, they will say, look for antioxidants. We buy them, supplements and all whatnot. Now, at this level, how do you liaise? Maybe, because you see, sometimes when we find out all these things, it's talked, because we don't know how to move further. There's need to extract these antioxidants. It might be to compound them into drugs and sell, as people do. Or it can be juice, it can be any form, so that people can be consuming and we make our money. That's the commercialization we are talking about. So if we can engage other professionals, maybe chemists or even chemical engineering, yes, or pharmacists, they can help us in extraction, because maybe we are not deeply into that field. So they can help us to extract the active ingredients there, and we compound them into something that is edible. So please, you can move further on that. Thank you very much.
Thank you very much. The next presenter. The next presenter should come, please. Okay. Okay. A review.
Good afternoon, panel of judges, ladies and gentlemen. My name is Michael Mary Lamzo, and I'm here to present a paper on a review on isolation and characterization of protein from avocado seed, processing methods, functional properties, and food applications. Next slide, please. These are my seminar outline. Next slide, please. There's growing demand for sustainable, low-cost alternative plant-based protein. As we all know, avocado seeds are underutilized. Most times when we get avocado, we just eat the pulp and throw away the seed. So there's like demand for protein from avocado, from plant-based materials. There's shift towards plant-based protein due to environmental and health concerns. Most people are trying to take more of plant proteins instead of animal proteins, because animal proteins are they don't digest easily compared to plant proteins, so there's shift towards utilization of plant proteins. There's increased interest in the agro-industrial byproducts as protein sources. Avocado seed is a major byproduct generated during avocado processing. Next slide, please. Next slide, sir. Avocado seed, which is about 50-35% of the fruit weight, contains 7.5 to 23% protein. It has high starch, dietary fiber, and bioactive compounds. It has potential for functional food formulations, nutraceutical applications, and supports waste valorization. Seeds are nutrient-dense but underutilized. Next slide, please. Plant proteins are derived from legumes, cereals, and fruits. Then they are important sources of dietary protein. They possess emulsifying, foaming, and gelation properties, increasingly used in sustainable food systems, and they support human nutrition and food security. Next slide, please. Next slide, sir. Oh, sorry, sir. Then animal proteins have higher digestibility, like I said the other time, and plant protein is easily to digest. That's why there's this shift towards, consumption of plant proteins. Then plant proteins are associated with lower risk of chronic diseases. They contain dietary fiber and antioxidants like the tannins, phytates, and then the lower environmental impact than animal proteins, are more sustainable and cost effective. Next slide, please. Okay. Process of avocado seed isolation. So the process of separating protein from other components, it improves protein concentration and functionality, enhances digestibility and food applications. Commonly used in food product development like the baking industry and the likes. Next slide, sir. Okay, the principles of avocado seed extraction. The cell wall disruption releases intercellular proteins. The protein solubilization occurs under suitable conditions. Solubility is affected by pH, temperature, and ionic strengths. Proteins recovered through precipitation and drying. Then functional properties must be preserved. Next slide, sir. Yes, so pretreatment of avocado seed. Why do we need to pretreat avocado seed? Avocado seed contains anti-nutrients like the tannins and phytates. So for us to reduce them, most of the time when they are taken in high quantity, it causes indigestibility and other health issues. So the pretreatment methods are advised to be used, like the soaking, boiling, and roasting, the drying, fermentation, defatting. Why do we defat? Avocado contains starch and other nutrients aside protein. So for the isolation process, it needs to be defatted and removed of any other nutrients so as to have high yield of protein from the material. So the benefits of avocado seed protein: improves protein accessibility, increases the pretreatment method. Benefits of the pretreatment methods: it improves protein accessibility, increases extraction efficiency, reduces nutritional factors, then enhances protein functionality, increases protein yield. Next slide, please, sir. Avocado seed protein extraction methods. The alkaline extraction method has been noted to be the most common and widely used because it is cheaper compared to the other methods of isolation. So imagine novel extraction methods: enzyme assisted and ultrasound assisted extraction, the microwave assisted extraction, and the high pressure processing membrane separation technology. Next slide, sir. The bioactive properties of avocado seed protein. The bioactive and nutraceutical components contain avocado seed protein contains flavonoids, phenolic compounds, antioxidants. So the health benefits of this seed protein: antioxidant activity, anti-inflammatory effect, antimicrobial properties, anti-cancer potential, cholesterol reduction, then cardiovascular health improvement. The next slide, sir. The functional properties of avocado seed protein: the solubility, influenced by pH, affected by temperature, dependent on ionic strength. Then the water absorption capacity, improves texture, enhances viscosity. Then the oil absorption capacity, enhances flavor retention, improves mouthfeel, taste. Then emulsifying properties, stabilizes oil water mixtures, useful in beverages and dressings. Then gelation properties, provides structural stability, important in meat analogs. Then the foaming capacity and stability, useful in cakes, ice creams, and whipped toppings. The next slide, sir. Yes, sir. So the applications of avocado seed protein, the food applications, can be used in bakery products like the bread, cakes, biscuits. The plant-based meat products like meat analogs and meat extenders. Then protein-enriched beverages like the sports drinks and smoothies. Then for functional foods, that's nutritional snacks and dietary supplements. Then industrial applications, in edible fumes application, biodegradable packaging materials, and nutraceutical products. The challenges and future prospects. The challenges is, if the first, low extraction yield, if the avocado seed material, the flour, is not properly defatted, or if the pre-treatment methods were not really followed, it can lead to low extraction yield. Then the presence of anti-nutritional factors. Avocado contains tannins, phytate, and others. That's why the pre-treatment method is important, to reduce it at least to the barest minimum. Then it has bitter taste. The pre-treatment method as well helps in the reduction of the bitter taste of the seed. Then lack of industrial scale technologies, limited safety and toxicity studies. The future prospect is, it's an advanced extraction technology. Like I was talking to someone one time, and he said, I was talking about isolation and characterization of protein. I was like, oh, that's a very good prospect in food science and technology of nowadays, because the world is shifting to plant proteins, and then isolation of it is a very good thing. So improved processing techniques, and then commercial scale productions. In conclusion, avocado seed is a promising alternative plant protein source. It possesses valuable nutritional, functional, and bioactive properties. Both conventional and emerging extraction techniques can recover quality proteins. Potential applications exist in bakery products, beverages, meat analogs, and packaging materials. The utilization of the seed protein promotes waste valorization. That is, when these waste products are used, there will be little waste, products around. Then food sustainability, environmental sustainability, development of functional foods. Next slide. These are my references. Thank you for listening.
Thank you so much. Wow. All of us, we have been eating avocados fruit and been throwing the seed away. Now we have learned that nothing about avocados is waste now. In short, I will not be surprised that the rind too will be useful for production of tea.
It's possible.
Yes. All these things, as I used to tell my students in the class, as a food scientist, if you are studying it and it's not even having impact in your own life, it does not even matter go away and study another thing. Because you cannot understand it better if you cannot put yourself and be the one people will be studying. Our food can be our drug if you put our mind into it. Avocado seed is a very useful seed in all ramifications. We use it to produce all this margarine at home, all these short things. We used to produce with it. Just get the avocado itself, add one thing or the other. I will not mention too many things, and blend. Use it to eat your bread. Use it to eat your yam. Use it to eat your rice without using vegetable or your pepper. Very interesting. Just a little lemon into it. Off you go. And your system, the probiotic system in your own body will be praying for you. All this sugar we consume, we don't even think on how we can neutralize those sugar. We don't. And we can still use the same food to turn it around and become positive for us. Diabetes. You are running from this, running from that food. You should learn how to balance your food. Because if you don't eat better food, you won't grow well. You won't grow well. Growing well does not mean you should be fat. You should know. Healthy is the word. Thank you for the presentation. Any question in the house? Yes. Please help me to give it to you.
Okay, thank you very much for the presentation. You've taught me one thing I've been searching for today. Because when these guys advertise the seed of avocado, they always tell you it lowers BP. And when you ask them how, they cannot explain. But you have just told us something now that the salt, because it drains the salt, that's why the BP drugs, some of them you urinate a lot. So thank you very much for that. Then my other advice is this. I saw in your challenges, you wrote a low product yield or something like that. Yes, you have to still work on that because any investor may be scared of putting his money down because of the yield. Because if you convince him that the yield can come up, they will bring money. So thank you very much. Well done.
Hello, sir. About the low yield she was mentioning, if you look at it very well, she was talking about the pre-treatment. The pre-treatment, that if you don't do the treatment very well, you may get low yield. That's what she's trying to say. And that one is germane in that work. Really, really germane. Yes, it will reduce fungal growth. Yeah. Yeah. Come on now. They don't pre-treat. So when you buy, you have to do it. You soak first. So the processing is not that it is hard, but we can actually do it. It's doable. Thank you very much. The next presenter, proximate composition and growth performance analysis on different street snacks fed to winning waster rats. Abel, have. Are they on ground? Yes, thank you, sir.
Good afternoon, the chairman, the fellow on seat, fellow food scientists, ladies and gentlemen. I am Abel Silas, here to present a seminar titled Proximate Composition and Growth Performance Analysis on Different Street Snacks Fed to Winley Worcester rats. Next slide, please. These are my presentation outline. Next slide, please. Introduction. Snacks are small portion of food generally eaten before meals or between regular meals, according to FAO 2011. And the same FAO classified snacks as street foods, ready-to-eat food and beverages prepared or sold by vendors or hawkers, especially in streets and other similar places. Olakpade and Awo, 2012. Next slide. Justification. There are serious wrong manufacturing practices associated with consumption of improperly produced, ready-to-eat street snacks. And when this is done, it has a negative impact on the health status of such community. It can lead to diarrhea, vomiting, and many other intestinal problems, according to Pikuda and Elelaboye, 2010 and 2014. Also, there is need to enforce good manufacturing practices. Recently, if you could remember, NIFST, through their podcast, June 6, there was a street food safety initiative, and it's on snacks. And if this is not done, we want to commend NIFST for this program, and we only pray and believe that there'll be more on this. Thank you very much. Next slide, please. Objectives of this study. We have major objective and specific objective. The major objective is to determine the proximate content of these snacks and check on by evaluating the group performance analysis. How do we go about this? Going through the specific objectives that are listed out are getting to do the proximate analysis of the snacks, assemble basic ingredients to formulate basic diet for the snack for the rats, carry out proximate analysis of the formulated diet, and assemble together these rats in a healthy environment. Then we carry out the group performance analysis by checking on the feed intake, the growth rate, and checking on the feed conversion ratio. Next slide, please. Materials and methods. This work was done under two phases. Phase 1, that's the sourcing of the snacks itself, and phase 2, the feeding trials. The feeding trials, we have some feed that are made for the rat chow we call it. These are the positive. Next slide, please. Experimental rats and management. 30 Winley Worcester rats were procured from loose physiology department, Idiaraba, Lagos here. Yes. And they were randomly selected and distributed along 5 feed treatment groups. What are these snacks? 1, we have chin chin, we have puff puff, we have rolled snacks, rolled biscuits, and we also have the last one, which is the peanut biscuit. Then the PCR is the rat chow that was fed to them as a positive control, with which we now monitor the other snacks. So we have 5 treatment groups of 6 rats each. That 5 by 6. And the animals were fed 150 grams of the body weight daily per group. Next slide, please. Next slide. Next slide, please. That's when we're in the lab. Next slide, the results and discussion. The proximate composition of the snacks were determined, and in table 1, these are the results. Next slide, please. Table 2. Table 2 talk about the growth performance characteristics of the rat. Here they were fed daily for 28 days. The feed intake, the growth performance of each of these diet group were taken every day. And after the day, the following morning, the feed that were left as remnants, we pack them, weigh them, and discard them. Feed another set of the weighted daily intake. And from the proximate composition in table 1, we can see that the moisture content was highest in PFP group, which is the puff group, at 20 years. And the least was the CFR, which is the rat chow, 11.21. The crude protein content was highest, and it fall within the average daily expected range of 17 to 19% crude protein. So here we have the highest at 17.2, which is marginally higher compared to the puff group at 14.14, CPC group at 16.8. Now, in table 2, that is the growth performance characteristics. Thank you, table 2. The initial weight fall, they are not significantly.
Which one is
CPC, that is the chin, the PFP, the pop, CPC is the chin, GFP is the, we have put it here. These are the legend. Then CFR is the control diet. I talk about the rat chow. The RFB, these are the rock bones. Now, each one of them were fed to the different treatment groups. And after 28 days, the growth performance were analyzed. Now, from the table, we can see that the final weight gain was highest, was significantly highest. Yes, at the highest, followed by the chin at 185.28 grams. And the last was the CFR. However, you can see at 169.01 grams, it was significantly lowest. Now, when you look at the daily feed intake of the 5 different groups, the daily feed intake was not significantly different from one another. Though we can see that they are marginally a bit different from one another, but significantly no. Now when I look at the feed conversion ratio, the feed conversion ratio was more beautiful in puff at 6.34, followed by chin, followed then by the rock bones, RFB, and the last, thank you very much, the CFR. And from what we can see here, mortality, there were no mortality. That is to tell you and me that very well. That's to tell us that the treatment group, the snacks that were fed, they were free from anything that can cause them ill health. The processing, the production were excellent from the table 1, and it showed in the group performance results. But you will see, please flip back to table 1. The moisture content of puff was very, very high at 28, and that showed during the study, because the products went bad after the 4th day. And like I said, we produce twice in a week because we cannot give them rancid puff. We cannot give them rancid food products and the rock bones. Though at this value, they still fall within some acceptable levels. Next slide, please. Now, the results of the proximate composition, I've already said this, range between this and this, and the moisture content of puff, like I said earlier, was too high, and it results in the product going rancid. Yes, after the 4th day, but from 5th day we can no more use it, and that's why we have to prepare another set with the same methodology. Next slide, please. Conclusion. The diets were palatable and easily acceptable to the rats because they were well formulated. And that is why we are now calling on our people. Thank God for our street food safety initiative. We should do more before the year runs out. We should let our people know, because the theme of this program is what? Harnessing our food science innovation. For what? For industrialization and economic what? Sustainability. If the economic is not sustainable, that's a problem. Then why are we gathering here? Our gathering here is of nonsense, of no use. So please, we still want to plead to our NIFST officer, the highest level, that we should do more advocacy programs and let the people, especially the vendors of these snacks, help us to have, we are making a special plea through this forum that we should make advocacy program and call people into order, run up and down, make noise as we used to. God will strengthen us the more. Thank you very much.
Thank you for your presentation, sir. Any question? Now everybody can eat pop-up shin shin now. Rat eats. Any question? Okay, please help me to pass the microphone.
Who is raising it?
Thank you for the presentation, Dr. Silas. Yes, I just want to, I don't know, the pop-off contributed to the weight of the blister rat more, probably because of the oil content. The oil in the pop-off will be higher than other products you have worked with. Do you agree? Did you do proximate composition? Okay, can we see the slide? Sorry, one John Lord. Okay. Okay, anyway, what I'm trying to point out is this. If we have to recommend that we should be, no, not this one. We should be eating.
For this presentation, we waited on the road performance.
Yes.
There are other results from this work outside the scope of this
Presentation. Exactly, I know.
the
Yes, sir. Okay.
From the digestibility study, yes, the pawpaw has this value, but he recorded the least of the digestibility, and that is to tell you and me that you have to be careful eating pawpaw.
We are going there together, and that is what I'm driving at. From the proximate composition here, we see that the crude fat content of pawpaw is even lower. I think if is more or less probably the second, and the presence of the trans fat we don't know, because the same amount, the same oil was used for the product, for all the products. I'm just looking at what could have contributed to that weight gain from eating pawpaw, and if that weight gain was recorded, the weight gain doubled within a month. So that means we run a risk of obesity and many other things coming up, especially amongst our children when they feed on this. Exactly. It has to be all encompassing. Okay. Thank you. The reason.
Thank you very much, sir. Thank you for that beautiful presentation. The next presenter. Hello, one house, decorum. Preliminary evaluation of blood pressure response to a developed antihypertensive. No. Okay. The last presenter. Yes. We are waiting. Mineral composition of complementary food from quality protein, maize and kidney beans by Dr. Adebayo Oyetoro. We are waiting for you, ma. Okay, you are standing in. Beautiful.
Good morning, ma. Good morning, sir. Good afternoon, sir. Good afternoon, ma. My name is Odebumi Babatunde. My name is Odebumi Babatunde. I'm presenting the topic on mineral composition of complementary food from quality protein maize, kidney bean blends. Next slide, sir. Introduction. Complementary feeding. Okay, sir. Complementary feeding. Immediately after 6 months, our babies, they need more than breast milk for them to develop more. So that is why we need to add solid or semi-solid food and to supplement their meal for their growing nutritional needs. Also, malnutrition in Nigeria. Malnutrition remains a significant public health challenge affecting children and the elderly. Also, we need to bring in nutrient-dense food, such as this QPM and kidney bean. Affordable, locally sourced ingredients are needed to prevent protein energy malnutrition, micronutrient deficiencies, and growth faltering. Next slide, please, sir. Next slide. So, why this study? high prevalence of mineral deficiencies among our infants and young children. There is need for affordable and locally sourced complementary foods, and underutilization of quality protein maize and kidney beans in infant food formulation, and also the potentials of cereal legume blend. Why this study? Potential of cereal legume blends to improve mineral intake and nutritional status. So the objectives of the study. The first objective is to process QPM and kidney beans into flour using standard methods for complementary food production. Another objective is to prepare 4 formulations, which the control will be 100% QPM, and other blends will be added to it. Also, to evaluate the mineral content of the complementary food samples using standard methods, and finally to determine the effect of increasing kidney bean inclusion, because quality protein maize will be the major nutrient, while kidney bean will be added in bits. Next slide, please. Quality protein maize enhance essential amino acid profile, especially lysine and tryptophan. Better protein quality. It supports growth, weight gain, and protein utilization, and it is the base ingredient in all the 4 formulations we'll be using in this study. Red kidney bean, it is rich in protein, dietary fiber, iron, and zinc. It has high mineral density. Soaking and boiling reduces the antinutritional factor, and we are going to be including it in this formulation at 10%, 20%, and 30% levels. Next slide, please. Hello, sir.
Next slide.
So, materials and methods. Our materials were gotten from reputable markets, and the production process was done in an hygienic manner and according to recognized standards. Those are the samples. We have 4 samples, as I said before. The first sample, which is the control, has quality protein maize as the 100% material, while the other samples are there as shown on. And the processing steps. For the kidney beans, it was soaked overnight and boiled to reduce the antinutritional factors in it. And both were dried and milled into flour. Blending was done according to the 4 formulations we used. Next slide. Mineral analysis was carried out according to AOC standard using AS. So the results. Mineral analysis of complementary food from QPM enriched with kidney beans. Potassium content increased by 145% from 29 to 71 milligram per 100 gram. Magnesium increased by 536% from 7.22 to 45.93. Calcium increased by 509% from 2.88 to 17.53, while phosphorus increased by 75%. We can see the results as shown on the slide. Next, this is the visual comparison. All minerals increase significantly with rising kidney bean inclusion. It shows that kidney bean have high mineral content. So the higher the kidney bean, the higher the mineral content. That is what the result is showing here. And the sample RDE, which has 30% kidney bean, consistently recorded the highest mineral values across all parameters. This shows that the statement that kidney beans has high amount of mineral is actually very true, because the sample that contains the highest amount of mineral of kidney bean had the highest mineral values across all parameters. Next slide, please. So, discussion of the results. Mineral enrichment effects. Kidney bean enrichment significantly improved all mineral parameters. As we've seen from the results, sodium rose from 23.8 to 51.75. Potassium the same, consistent with kidney beans' high mineral density, as recorded by Adeyeye et al. In 2022. Also, infant health relevant. Elevated potassium, calcium, magnesium, and phosphorus levels are critical for bone development, energy metabolism, enzymatic activities, and overall growth in infants and young children, as recorded by Adeyeye et al. 2022. Also, this work in line with the literatures. Findings align with prior cereal legume blend studies showing improved mineral composition in complementary food. According to Bello et al. 2023, Nkata et al. 2018, fermentation and germination can further enhance bioavailability of all these minerals in our diet. And finally, all differences between sample groups were statistically significant, p less than 0.05, with distinct superscripts grouping each mineral column, validating the dose-dependent effect of kidney bean inclusion. Next slide, sir. Conclusion: quality protein maize and kidney bean complementary foods were successfully developed in 4 formulations. All 5 minerals increased significantly with rising kidney bean inclusion. Sample RDE, which contains 70% quality protein maize and 30% kidney bean, had the highest mineral contents across all parameters. This type of blend, cereal legume blending, is an effective, affordable strategy to improve infant food mineral density. And finally, these formulations have potential to address micronutrient deficiencies among infants and young children in Nigeria. So further processing is recommended, such as fermentation, germination, in order to further should be explored to enhance mineral bioavailability. Also, anti-nutrient analysis. We all know that kidney bean is a legume and it contains anti-nutritional factors. So there should be what? Analysis on anti-nutrients like phytate, saponin, tannin, so as to what? Assess the level of anti-nutrients in the diet. Also, sensory evaluation. Consumer acceptability trials should be conducted with mothers and caregivers. And finally, bioavailability studies. In vitro and in vivo bioavailability studies are needed to confirm mineral utilization. Thank you. These are my references. Yes.
Thank you for the presentation. Do you have questions? Yes, ma'am.
Thank you for the presentation. My question is, of all the blends, what will be the recommended one to use? Because it may not necessarily be the highest, because all these minerals, we have a required intake that we should take. The fact that it is the highest does not mean it is the best, because if we take too much of these minerals also, they also have the side effect. So I think this study should go further and tell us which of the combination, which is the best or the premium one to take. Thank you.
Thank you very much for the presentation. Is a comment I want to make, and it's not limited to your presentation alone. You know, all these legumes and the things we've been trying to blend to get something. What I keep asking myself, what is the availability of the raw materials? Because you see, that is one challenge we food processors have. And when you talk of commercialization, you have to think of food manufacturing, not limiting only to processing. So the availability of these products is always a challenge. You know, you can't talk of, if you talk of cassava or palm oil or granite, you know, those ones, we've really excelled in their production. What do we do? Because sometimes I said, should we leave food processing and go back to agriculture to have our supply? Because any serious investor, as I keep saying, will look at it. Can it be sustainable? If I build my factory here, where will I be getting all this high quality maize and the kidney bean? So, so please let the research also go back to that backward integration, so that it will be more convincing. Thank you.
Good afternoon, everyone. I want to add, make a comment concerning what he just said about availability. It is important that the produce is available. It is true what you said, but we also are running into a situation whereby some of these nutritious produce are going into extinction. And so while we consider availability of raw material, it's also important to emphasize that these crops, the nutritional properties of these crops, because at the long run, you are also helping availability of that crop. Because creating that awareness sends the farmers back to the farmland, and because of its importance, more of it will be produced. So I still encourage students to research the underutilized crops.
Thank you.
I just want to make a very quick comment. I want to commend the team that is working on this topic. And the presentation of your slides is fantastic. I give it to you. It's colorful, and it's it speaks to the topic, and the audience is able to follow you. So apart from what they have said about the availability of the kidney beans, you could go back, it's a pity that the whole people that made their submission today, that presented, nobody presented on cassava, which we are so rich in Nigeria. So are working on cassava. So we are moving into innovation now. Okay, so thank you so much. Please keep it up.
Thank you for your presentation. Lastly, before we close for today, I want to shed more light on the issue of availability. Actually, decorum, please. Actually, that word, availability, it is subject to what we want. In Lagos here, nothing is cheap. If you want to cite your company in Lagos State, I'm very sure, I don't think we have a very large farm in Lagos that can give everybody. That is one. So when you talk about availability, you are in Lagos, nothing is available for us. That is a fact. Because all of my project work, if you look at my works, they are all on underutilized grains, cereals, crops, even plants, underutilized ones. That's what I always run after. It's because we want to make awareness so that we don't overflog those ones that we have, leaving those ones we have not been using at all to go on extinction. Now, we are talking about pigeon pea today. We have almost, in short, I was working with pigeon pea with Dr. Balaji. I was surprised to note that pigeon pea and this second African yam bean, they look so similar that we have to be sending it back to IITA for identification, so that we are not going to be giving false information to the public. And now, how much do we buy it? But here in Lagos, we never find it. And if we find it in Lagos, it will be times 10 of what you are going to buy it from where they are producing it. Are we going to tell those ones that it's not available? It's available with them. It is we that we don't have it. So what I want to say is, once we want to work with anything, we should try and look for where is it more abundant. Link with them. Thank God we can transfer through vehicle, through anything now without moving our body. So like that, there will be less talk on availability. If you help those farmers, help those ones, they will know that we need it in Lagos. They will take it upon themselves, bring it down to us. But if you don't try and make projects out of it like this and people begin to look into them, they will not know that it is needful and they will keep farming. That is what I observed during these years of my experiments. So availability is not a general term. It's not a general term. All these things are available where they are producing them in abundance. Even it is wasting. It is wasting. The farmers are crying that we will give you, go and work. If they start to sell it, we give you money. To that extent. So please, let's start and look into all these our local materials. We are blessed, and I know we shall be more blessed in Jesus' name. Well, we have come to the end of the presentation for today on this our side. I don't know whether they are true over there. They are true too. So we thank God for the good presentation. For me, is the person ready now? Okay. Are you going to be needing the microphone?
Okay. Good afternoon, everybody. Good afternoon. My name is Ayeni Elizabeth. I'm here to present the topic, sensory evaluation of baked kokoro produced from blends of maize flour and fiber-rich fruit pulp, orange fruit pulp. Next slide. Next slide, sir. Sir. Okay. Nigeria produces large quantities of citrus fruits generally, especially oranges. And you will agree with me that after the extraction of the fruit juice itself, we are left with the peel and the seed to be discarded. So over time, we discovered that the pulp is being thrown out into open spaces, and it causes environmental pollution, and then it blocks our drainage system, and then you have waste. So, and this is why we got interested in the project to use the pulp as a blend in the mixture for the production of cocoa. Juice processing generates significant byproducts, which I just mentioned: the pulp, the peel, and the seed. Orange pulp waste in Nigeria, according to Pacheco, Menero, and Vine Limited 2019. Poor waste management leads to environmental issues, like I mentioned. They are being thrown into especially if you go to places like My 12, you go to Ile market, you will see where they deposit the waste from oranges. And then industry too contributes to this by discarding them in drainages, open spaces, and all that. So there is need for sustainability, sustainable utilization of agro waste. Indigenous snacks in Lagos, in Nigeria, kokoro, for example, is an indigenous snack. And then we have limitations of traditional kokoro. It's produced from maize, and then we have it's limited. It's carbohydrate-based food, and it's limited to the carbohydrates that we get in it, and then we don't have enough fiber in it. Next slide, please. Okay, statement of problem. Only about 40% citrus fruit is used for juice production. And what do we have? We have the other parts being as waste. About 24.3 million tons of citrus waste in general annually. According to the picture, you can see that in an open space, they are being discarded and all that. Limitations of traditional cocoa. Like I said, but it's not everybody that can eat cocoa because of the health benefit. And then it has lower dietary fiber So justification, orange pop flour as a solution. Why do we choose orange pop flour? Yes. So what we did, orange pop flour. The orange pop, we all know the pop. After juicing orange, we have the pop that we normally discard. So we find that it has much health benefit. It has dietary fiber. It increases gut health. So, and that is what we use in the blends for the kokoro production. Functional benefits of orange pop, I've mentioned that, and of course environmental benefits. It has environmental benefits because we will be discarding that into the environment as well. Next slide. Okay, the aim of this study is to evaluate the sensory attributes of baked kokoro produced from blends of maize flour and fiber-rich sweet orange pop. For the kokoro that we all know is usually being fried, but in this sense we are baking it. We want to reduce the fat content of it so that it can be consumed by everybody, even people living with diabetics. Objective of this study is, the specific objective of this study are to produce number one, orange pop flour. Now, I know we all understand this, making flour out of the orange pulp that we normally discard out there. So it is being dried. When we did this, we dry it, and then we have our flour. Then we mix into the blends of your normal already blended maize. Now, to produce maize flour, of course, we are all used to that. And then produce kokoro with the varying blends of maize flour and fiber-rich sweet orange pulp flour. Evaluate, and then we carried out a sensory evaluation on that, which is our major topic. Next slide. Next slide, sir. Okay, methodology, we have the raw material selection, sweet orange and maize. And then dry maize and mature old grains. Old orange were purchased at Ogere markets in Ogun State. And then the equipment we use, we gotten from the lab of the food technology department of Gateway Polytechnic. And then these are the formulations. For A2, we have 250 grams of maize, and we have fiber-rich sweet orange pulp flour. That's the flour we got from the orange pulp. That's A2 is our control, which is 0 gram of that. So we have purely the maize. We have B2, which is 45 grams of the flour, and then we have 5 grams of the orange pulp flour. And then for C2 sample, C2, we have 240 grams of maize and 10 grams of the orange pulp flour. And then we have D2, 235 grams of maize and 15 grams of fiber-rich orange pulp flour. And then E2, we have 230 grams of maize and 20 grams of orange pulp flour. Sir, next slide, sir. And this is the flow chart, then the procedure for production. The maize was gotten and then with salt and then wash in the potable water, and then it's dried. Next slide, okay. And this is for the production of our orange pulp flour. Okay, sir. Next slide, sir. The results. Okay, the statistical analysis was carried out. Next slide, result. Okay, here is the result. Under here, we'll discover, if, like we all see, for sample A, we have the appearance to be this figure 5.64 plus or minus 1.04, whatever. So overall acceptability, sample B has the overall acceptability, as we all see. From the appearance, according to sensory evaluation, from the appearance to the texture, the taste, aroma, and crunchiness, overall acceptability, we have sample B2. So in conclusion, incorporation of the fiber-rich sweet orange pulp flour up to 5 grams which is in bracket 12.5%, enhances the sensory quality of the baked kokoro, and that is what we're trying to achieve. Instead of eating your normal kokoro that way, and then you have something that can increase fiber, your dietary fiber, and is acceptable as well. So the study demonstrates the orange pulp flour can be effectively used to improve product quality, not only the kokoro itself. And it reduces the citrus waste. Of course, we are trying to reduce waste and promote sustainability of raw material utilization. And then therefore, we have a 12.5% substitution level of fiber-rich sweet orange pulp flour into maize flour. We recommend that to you. Thank you for listening. And these are my references. Who will bring it? I will bring it to the NIFST conference. Yes, yes, ma. Yes, ma. Tomorrow, NIFST conference in October, ma. The white maize was used, yes.
Because I know actually for this time. Did you fry?
After getting the blends of the maize flour, we add little oil so that it's not be sticky, and then we put water inside pots. While it's boiled, we add. Yes. So what we did was to we steam like a kind of cool. We pour the dried powdered maize inside it, and we continue to stir. We stir until we have a thick dough-like. Then we roll it out, then cut, and then we bake.
So it will not stick. So they will first gelatinize. That is what she just explained. Yes. But the dough, just to gelatinize it so that it will form, it won't be breaking. So you are bringing the kokoro tomorrow. It's you that say it, oh. It's not me, oh. Okay, no problem. Thank you for the presentation. So another use of our orange rind, which means it's not limited. The pop. That white one. Okay, all those. You now dried it and make it to flour. Nothing is a waste again. Waste to wealth. Here we come. Àtàwọn tinú ẹ̀ tá máa ń jẹ yẹn. For those of us that used to eat the inu, we can eat that and use the white to do this. It's still good. Nothing is a waste. Because that one still have pectin. The white, yes, it's eatable. And it's very good. It moves the bowel very well. It's the fiber. Thank you for your presentation. Any question? No question. Thank you very much. On this note, we draw the curtain.
Group photograph. Announce for group photograph.
Okay. We are questions. Where? Come forward, sir.
Good afternoon, Zumas and Sars. My name is Okekunle Hafiz Damlari. I want to ask about the orange. Since you got the raw material orange, are you going to go into production of juice? Because for mass production to be expensive to get orange and extract only that pulp.
Yes. I will answer him. There are some companies that are producing juice from all those oranges. So all those pop is a problem to them. It's a waste. In short, one company just called me now to come and look into their waste so that we can turn the waste to wealth. So something like that could come up. Apart from just giving it to animal to eat up, we can convert it to something that is will be useful for man, and we are all good. So on this note, we draw the curtain. Thank you very much, ma'am. Do not stop. Go ahead. Put more light in your laboratory and continue working. So for me, as a person, I am privileged to share this session, and I did not only share for sharing sake or sitting down. I am blessed. I get more knowledge. I get more insight into what I am doing, that I am on the right path. Yes, I'm going home to give my students more information on what to do more to move to the next level. By this time next year, will be more than this, because many people are coming because they are going to improve on themselves. Thank you for coming. Thank you for your time. Thank you for your patience. More of us, all of us that are seated here, I'm expecting us to submit paper next year. Yes. Research institutes, university, polytechnics, individuals, government bodies, you can submit paper. Yeah. And I'm saying this now because we are going to the next level. We want our publication, NIFST, to begin to function now. When NIFST starts to function, it will pay everybody. All these things, it will not be a thing of in-house for NIFST alone. The publication will go outside the country, and people will see our ideology. They will call on us because your name will be there. And from there, you sell your information. So please, let's come up, and God will continue to bless all of us. Thank you, and God bless us all.
So thank you so much, the panel chairs, Dr. Okafor and Dr. Abegunde. And I cannot thank enough our audience. Thank you for staying with us till the end. Yorubas will say that Ibere ko nse oníṣẹ́, he that endure till the end, the same shall be saved. So I know that everybody you have learnt. So that's the reward of your staying till now. So we look forward to seeing you tomorrow, because that's the party. Today is academics, tomorrow is party. You have learned, so come tomorrow and come and boogie dance. The chairman, Lagos chapter, said she wants to say one or two things.
I just wanted to appreciate everyone once again. Thank you all for coming. Our chairman of the breakout sessions, thank you very much. All our elders and fellows in the house, Dr. Eshie, thank you very much for coming. Dr. Ladumoye, everyone, thank you very much. Our SON madam is here. She came in while we were about to run. I hope she will come early tomorrow so that she will not miss the show. Mrs. Aaron Ilewa, thank you very much for coming. Our madam from NAFDAC, thank you very much. We are really glad to have you. And just to let us know that the Innovation Challenge is on towards the national conference. Come up with your innovative food products. Check the platform, the NIFST platform, for all that you need to know about that challenge and put in for it. You can be the one that will win that award. And it's not small money. I won't tell you the amount. Go and check the flyer. Go and check the flyer. So many goodies are really, help. So thank you very much, everyone. Tomorrow we are starting, by the grace of God, on the dot of 9 with the debate. With the school debate. So please don't miss it. It's going to be highly contested. So please, let's come around. Tomorrow is going to be a great day by God. The rain could not stop us today. Thank God for that. Thank you everyone for despite the rain to still be here. God bless you all, and see you tomorrow by the grace of God. Thank you, our editorial madam. No, please, a round of applause for Dr. Yetoro. Thank you very, very much. For Adebayo Yetoro, your husband will not be annoyed. Dr. Abiodun Adebayo Oyetoro, complete. The same way we had Dr. Wabweze Peter. Oh, God. Thank you very much.
So please, when you are coming tomorrow, make sure you come with your tags. We are not going to produce another one for you. And this is your ID. If you don't come with it, We won't allow you to come in. Yes, please come with your tags. So on that note, we want to appreciate you once again and see you all tomorrow by the grace of God. I wish you safe trip back to your respective homes.